Now we're gonna make a little bit deep diving in the mechanism of actions for the chemotherapy different categories. First, we should discuss some basic informations about the DNA and its components before the start of talking about the mechanism of action of chemotherapy, such as antimetabolites. First, we should know more about nucleotides. A nucleotide consists of 5-sided sugar molecule, either ribose in RNA or deoxyribose in DNA, attached to phosphate group and a nitrogen containing base. As you can see here is a 5-sided sugar molecule and attached to the nitrogenous base. And here is the phosphate group. The bases used in DNA are adenine, cytosine, guanine, and thymine. This is the structure of the deoxyribonucleic acid DNA. Here adenine, thymine, cytosine, guanine. In RNA, the base uracil takes the place of thymine. It's the basic structural unit and the building block of DNA. These building blocks are hooked together to form a chain of DNA, as you can see here in this graph. The purine base A and G, or adenine and guanine. The purine base A and G, here is the structure of the adenine and guanine in the DNA structure. As you can see here, adenine, which is paired with hydrogen bond to thymine, and guanine, which is paired by hydrogen bond to cytosine to form this DNA structure. Here is the pyrimidine base C and T, cytosine, as you can see here, and thymine. And here is thymine, which is paired by hydrogen bond to adenine, and cytosine, which is paired here with guanine. This slide is about the mnemonic regarding purine and pyrimidine. Purine, pure as gold, adenine, and or guanine. And when it comes to pyrimidine, we should remember this statement: cut the pie, C for cytosine, U for uridine, and T for thymine. DNA structure, as you can see here, deoxyribonucleic acid. Here is the major groove and the minor groove. And here is the sugar phosphate backbone and the base pair between adenine, thymine, adenine, cytosine, guanine, cytosine. And here's the structure of the nucleotide. For example, for the adenine with its nitrogenous base and the sugar, and here is the phosphate group. Purines always pairs with pyrimidines. As you can see here is purine, or pure as gold. Its member is adenine, and guanine is paired with cut the pie, which is cytosine and thymine. Purines always pairs with pyrimidines. It is a crucial statement. Purines always pairs with pyrimidines. Here is the base pairs: thymine, adenine, cytosine, guanine, to form the deoxyribonucleic acid. Now we're going to start the antimetabolite agents, mechanism of actions. Antimetabolites, S phase. Antimetabolites are chemotherapy drugs that prevent cancer cells from making more cancer cells, replicating. They trick cancer cells into using the drug instead of molecules it needs to make the genetic material to replicate, or DNA. We should know that thymidine monophosphate, or TMP, also known as thymidyllic acid conjugate base thymidylate, deoxythymidine monophosphate, or DTMP, or deoxythymidyllic acid conjugate base deoxythymidylate. DTMP, deoxythymidine monophosphate, consists of a phosphate group and pentose sugar, and the pentose sugar deoxyribose, and the nucleobase thymine, which is a pivotal part of the DTMP. Mechanism of action of methotrexate and the effect of leucovorin. As you can see here, the diet or intestinal flora produce folate, and the folate enters the cell or the target cell. Inside the cell, the folate converted to the FH2, or the dihydrofolate, which is converted to. And as you can see here, the dihydrofolate reductase enzyme catalyzes the conversion of folate to dihydrofolate, and after that to tetrahydrofolate. After that, the tetrahydrofolate is converted to N5N10 methylene tetrahydrofolate, which is thymidylate or deoxythymidine monophosphate biosynthesis, plays an essential and exclusive function in DNA synthesis and proper cell division. Thymidylate synthase catalyzes a crucial step in DNA synthesis by methylating deoxyuridine monophosphate to deoxythymidine monophosphate and using the N5N10 methylene tetrahydrofolate, and the deoxythymidine monophosphate is subsequently phosphorylated to produce thymidine triphosphate, DTTP, which is a vital precursor for DNA replication and repair. When it comes to the mechanism of action of methotrexate, methotrexate enters the cell through active transport process, and methotrexate inhibits dihydrofolate reductase, and as a result of that impair the synthesis of deoxythymidine monophosphate and inhibits DNA replication and repair. The N5N10 methylene tetrahydrofolate is crucial to form adenine, guanine, thymidine, methionine, and serine. Leucovorin rescue. Leucovorin or folinic acid acts as an antidote, functions to limit the myelosuppression for the GIT toxicity, nephrotoxicity, and the neurotoxicity result as secondary to high dosage of methotrexate and other folic acid antagonists. It should be administered 24 hours after methotrexate dose. Administer N5-formyl tetrahydrofolate or leucovorin or folinic acid, which is converted to N5N10 methylene tetrahydrofolate, and therefore bypass the inhibited reductase mechanism by the methotrexate. Tissue toxicity may be permanent if the leucovorin therapy gets delayed beyond 40 hours. Mechanism of action of 5-fluorouracil. 5-fluorouracil, as you can see, enters the target cell and is converted inside the cell to the 5-deoxyuridine monophosphate, which competes with deoxyuridine monophosphate for the enzyme thymidylate synthetase. And as a result of that, the deoxythymidine monophosphate formation is inhibited. The DTMP is needed for the DNA synthesis and the cell growth. Metabolic pathway of cabazitaben to 5-fluorouracil. As you can see here, the cabazitaben as a prodrug is converted to the 5-fluorouracil. Three enzymes are involved in this conversion: carboxyl esterase, cytidine deaminase, thymidine phosphorylase. Mechanism of action of 6-mercaptopurine. 6-mercaptopurine is purine antagonist which inhibits DNA and RNA synthesis and acts as false metabolite, which is incorporated into DNA and RNA and eventually inhibiting their synthesis. As you can see here, 6-mercaptopurine is converted to 6-thioinosanic acid or TIMP, which inhibits the formation of the IMP or xanthosine monophosphate, which is converted to two important components, AMP and XMP. When it comes to the AMP or adenosine monophosphate, which is interconverted to ADP or adenosine diphosphate or ATP or adenosine triphosphate, and AMP is also a component in the synthesis of RNA, and AMP is present in all known forms of life. The IMP dehydrogenase or inosine 5-monophosphate dehydrogenase plays a key role in the biosynthesis pathway of purine nucleotides by catalyzing the conversion of IMP to xanthosine monophosphate or XMP, which in turn serves as a substrate for the production of guanosine monophosphate or GMP by GMP synthetase. Mechanism of action of gemcitabine. First, we should know about the DNA polymerase and ribonucleotide reductase enzymes. When it comes to the DNA polymerase, they are enzymes that create DNA molecules by assembling nucleotides, the building blocks of DNA, and are essential to DNA replication and usually work in birds to create two identical DNA strands from one original DNA molecule. And the ribo And the ribonucleotide reductase, or RNR, is the enzyme provides the precursors needed for both synthesis and repair of the DNA. For the mechanism of action of the gemcitabine, it inhibits the DNA synthesis by inhibition of DNA polymerase and ribonucleotide reductase. The phosphorylated active metabolites gemcitabine diphosphate and gemcitabine triphosphate. Gemcitabine diphosphate inhibits DNA synthesis by inhibition of ribonucleotide reductase, and gemcitabine triphosphate incorporates into DNA and inhibits DNA polymerase, and as a result of that inhibition of the DNA synthesis. Antimetabolites S-phase, capecitabine, fluorouracil. Mechanism of action: pyrimidine analog, inhibits thymidylate synthase, which depletes thymidine needed for DNA and RNA synthesis. Capecitabine is an oral prodrug of 5-FU. Fluoxuridine, analog of 5-fluorouracil. Gemcitabine and cytarabine. Pyrimidine analog, which inhibits DNA polymerase and causes chain termination, causes DNA breakage. Fludarabine, adenosine analog, which inhibits DNA polymerase when incorporated into DNA, causes DNA breakage. Methotrexate, folate analog, and inhibits dihydrofolate reductase, depletes purine and thymidine nucleotides necessary for DNA synthesis. Leucovorin folinic acid, used for reversal of methotrexate toxicity and as a part of chemotherapy regimens. Hydroxyurea inhibits ribonucleotide reductase necessary for DNA synthesis. Cladribine, adenosine analog, causes DNA breakage. Azathioprine, 6-mercaptopurine, 6-thioguanine, inhibits purine nucleotide synthesis.